mold chances?
How many days?
A month.
Okay. That seems like it could be. Oh, someone else just joined. Who this?
What?
Sophia.
Oh, Sophia. Yay. Hi, Sophia. Sophia's just the silent partner here. Okay. All right, people. Let's do this thing. So first, I want to start. This is actually pretty cool. Again, like if you can just mute unless you have a question. Really, since we're doing this live thing, go ahead and ask questions. Okay, that's the purpose of us being here together. So I'm here to answer your questions. Today's a bit of a small, like a shorter lesson, but it'll be a good chance to go over a couple things. So, oh, damn it! Of course I didn't do that. If you have your phone nearby, I'm going to forward you a couple of things that Richard just sent me. That are a couple of good questions. Okay, and I can actually open them on the screen share too. So he asked about that one. He said, what's the deal with this basically? How come that sugar is called a hexose sugar? Okay, I'm gonna start screen sharing, okay? Does everybody see this?
Yep.
Yeah, okay. So his question is, this says fructose is another hexose sugar. And then he sent that image and he said, which one of these two is wrong?
What image?
The picture, I sent another picture to you just now. So he sent the, like this one, the molecular.
Is it hectose or pentose?
It says that fructose is another hexose sugar. And then this is the structure of fructose. So his question is, isn't this pentose? And his question is actually not that. His question is, which one is wrong? Is fructose a hexose or is fructose a pentose? That's the question.
It's a hexose because it has 1, 2, 3, 4, 5, 6 carbon.
Where? And the picture is pentose.
So the shape of the molecule is a pentagon.
Doesn't matter.
Yes, exactly. That's correct.
It's the number of carbons.
Correct. So it's not based on the shape. So yes, you're right. Even in the slide here, it's actually, let's go here. Right. So here's fructose, right? So yes, it has a pentagon shape like this, but it has 1, 2, 3, 4, 5, 6 carbons. Where's that annotation thing? Here. Draw. Not that. 1, 2, 3, 4, 5, 6. So therefore, it is still a hexose. That means this is also hexose. And this is also hexose. These are pentose because they have 1, 2, 3, 4, 5. Right? 5 carbons. Richard, does that make sense? Richard, does that make sense?
Yes.
Yes? Good?
I think he said yes.
I thought I heard something. Okay, let's clear that. Okay, and then he had another question, which is about, oops, stop annotating, which is about this slide, okay, which has a mistake in it. That's a good observation. Okay, there's a mistake here. You can see that, let's take a different color, okay. Go ahead, Richard. If do you want to wait, do you know how to annotate or never mind? I can do it.
Then just go.
He was pointing out that this part that's highlighted takes an H and an OH, which gives us water. Yes, that's correct. But then somehow they were left with, if you look here, there's an OH and an H that's left, and then somehow it magically became H and H, which is a mistake. That's not how that works. The bond, let's clear this, the bond should actually form between these two, where one of them leaves one of the O's behind, right? So you take, get rid of that. You should actually draw it like this, where you get H, and then the whole OH of this one gets taken off, right? H2 and O, and then the O from this becomes the O here. This O goes there. Whoa. See how that works?
Then if you do this, wouldn't that like H, and then this O goes here? Isn't that the same thing?
No, because this H is actually attached to the O.
Oh, yeah, okay.
Yeah, so that doesn't work. It has to come off of this OH, off the edge.
Oh, okay.
Mr. Newland,
Yeah.
like when making maltose from glucose and glucose, is it alpha D glucose or beta D glucose?
Let's go check.
Up, down, up, down. That is beta.
Where did it go? No. Maltose, there. Yeah, so let's take a look. What's this?
Down, down, up, down.
Yeah, so then?
Alpha, isn't it?
Yeah.
But the one just now there, isn't it beta glucose?
Up, down, correct. Which is probably why there's a problem with this. Because if they had done this right, then this, then if they had drawn this correctly, they would have drawn this OH down here, and then they would have had OH circling the OH, and then they would have drawn it properly. So that's the issue. I think this is showing, this should not be beta.
Also, the mistake is this is drawn wrongly to a beta glucose instead.
Right, correct.
Like it should originally be alpha.
Correct.
Okay.
Should be alpha. Yeah. Good questions, people. Okay, anything else before we wrap up the proteins topic? Okay. All right then. I'm gonna get rid of this annotation. Let's close this. And I'm gonna show you on iLearn, you'll be able to download, and this will be kind of like a good, whoops, I don't know. iLearn is gone. Okay, great. Okay.
Those pictures to there.
You'll find, I don't know, I just kept it. You'll find this table. Okay, so essentially the end of today is you're gonna come up with this whole list of specific names of proteins. So I recommend filling this out once you're done. There is like a specific slide for every single one of these, so you can just kind of consolidate the information from there. But this is what you should probably use and kind of follow along with either now or later on. Okay, just what type of protein is it and what is the function? Okay, whoa, hello. Oh yes, don't forget about our YouTube channel. Follow, subscribe, like.
It's not a notification button.
Yo, what?
Jay's face is the
Yeah,
thing.
by the way, I didn't choose this. This is what just came up on its own
Wait,
by
really?
YouTube. Like, boom. That's what happened. Okay.
Like, subscribe, turn
like,
on
subscribe.
notification.
Yeah.
Subscribe.
And I'll see you in the next episode.
Okay, so let's go. Where's my PowerPoint? Here we go. All right, let's do this, people. So last time we had a little explanation of the 4 levels of structure for polypeptides, right? Oh yeah, that works. Even though it's only HL level, but that's fine. It's good to know. Okay. Here's where we're gonna pick up. So like we mentioned yesterday, there are kind of 2 types of proteins that exist, not 2 types, but like 2 ways in which proteins exist. So you can have fibrous proteins. I kind of like this. Check this out. Whoa. Whoa. Nope, don't do that. Okay. You can have fibrous proteins and you can have globular proteins. So fibrous proteins are proteins that are left in their kind of secondary structure. So they're like longer, stretched out proteins. They're usually there for a structural function. Okay. So examples like, I think it's easier if you just look at the example, things like keratin, which you find in hair and nails, fibrin, which we know from last year as, where do we find fibrin, or what's fibrin?
Veggie.
Platelet.
Fibrin. Where have you heard fibrin?
I think it's in
In
the
bio.
blood plasma.
Blood. Yeah.
Blood
What does it
clot.
do though?
Oh,
Sorry.
so like changes into fibrinogen, I think.
Oh, it's for the clot.
Wait,
Oh, wait, are you sure?
Blood clot.
Flip it.
Mhm.
William,
Nice.
flip it.
I'm confused.
Fibrinogen becomes,
Fibrin.
yeah,
Fiber.
right, in blood
Oh, okay.
clotting, right, blood clotting.
Oh, I was correct.
Yeah, I mean,
Like just opposite.
correct.
Oh,
Fibrinogen
I
is.
thought I said something like completely different.
Oh no, I was just telling you to flip it. So these are mostly structural types of proteins, okay? Whereas globular proteins take on the 3D structure, and they're usually things like enzymes or other proteins that have like a functionality, like they actually like affect a reaction or they transport something, but they're not just there for stability and structure, okay? One of the interesting parts that you can see here is that fibrous protein, why are you doing that? Oh, here we go. No, it won't let me do that. Okay, anyway. Oh, there. Do you have this, I don't need that, that's annoying. I'm just not gonna bother. You have repetitive, drawing is nicer actually. Repetitive amino acid sequence. So you have like the same amino acid sequence over and over and over, which makes it like this longer protein, right? Versus irregular amino acid sequence, which means you have like all like random mixture combination, which is likelier to just fold up into a crazy glob, right? So from last time, if we remind ourselves of here, that primary structure is the amino acid sequence, so that determines further structures. So that means if the primary sequence is repetitive, you're likely going to just end up with a secondary structure, a longer molecule. If it's more random and all over the place, you'll get more of a globular structure. Okay? So 2 types of proteins, fibrous versus globular. Their function is related to that. Yes, Joseph.
So for you, understanding 5, SL don't need to know this, right? Only for.
So the understanding you do need to know. It's the content on the slide you don't need to know. So understanding 5 just says you might have one chain, or it might be like many chains together. That in itself is not the levels, but it's just something to know that proteins can be complicated. They can be like multiple chains altogether, or they just could be one. but you don't need to know necessarily like what is each of the 4 levels about.
Okay.
Okay, so what we're going to do with the rest of this is to look at the versatility of proteins. So here you've got almost an endless list of things that proteins do. In fact, when we looked at membranes, we saw, remember Tracie, right? Tracie, Tracie, was all the different types of membrane proteins that exist, or what they could do. So here you can see some of them again. Here's membrane transport, for example. You've got receptors. There's not that many more from that group, but that group could also fit into this list. Here you have like all other things that proteins can do for us, okay? A little bit of a description. So these would be like a category, cell adhesion, right? Cells sticking together. But the ones over here in red on the right-hand side, the key examples, you do need to know these examples, and they're the same ones from your, that table that I just showed you. Okay, so that's why I want you to like fill out what their function is. For specifically 1, 2, 3, 4, 5, and then there's one more for spider silk. Okay, so very quickly, we're just gonna go through each of these 5 examples and just give a brief description of what it's for, where it's found, what its uses are, etc. Okay, so like I said, key examples outlined in more detail coming up. I'm gonna skip the biotechnology part for now.
What's spider silk?
Like spiders, silk. Like,
Why do we need to know that?
because it's a protein and it's cool. You'll see. Okay, let's begin. So the first protein is an enzyme. So the example that they are asking you to be familiar with is called Rubisco. The full name of Rubisco is ribulose bisphosphate. This is an enzyme that is absolutely critical for photosynthesis. Yeah, give me one sec. It is, if you look here, the reaction that fixes carbon from the atmosphere. So this is the enzyme that's really involved in taking the atmospheric CO2 and attaching it to an organic molecule and making that carbon a part of what will eventually become glucose. Okay, HLs, this is going to soon become one of your best friends. Okay. Yes, Michael, question.
We need to know the full name.
Since you're going to continue taking HL, yes.
How about SL?
You don't have to worry about that, right?
Jesus.
Then how about SL?
SL, you can call it Rubisco.
Okay, thank you.
Ribulose biphosphate. There's two phosphate in ribulose.
Correct, exactly. Okay, so Rubisco, very cool enzyme, very important enzyme for life on the planet. All right, this one you know, insulin. Can someone tell me what is insulin for based on IG knowledge?
Medicine.
I can't hear Richard very well. I think
Said
he-
medicine,
Richard's like, no.
right? He said medicine, I think.
he said medicine. Come on,
I
man.
don't know. That's what I heard.
We can do better than that. What's insulin for?
Is it
Pig.
to make something faster or something?
Or diabetes.
Control blood sugar levels.
Blood sugar levels, yes.
Hey Siri, what is insulin?
Okay, I found this on the web. What is insulin? Check it out.
Blood sugar levels is correct. When does your body produce more insulin? How about that?
When
It
too
promotes
high.
the absorption of carbohydrate.
Shush, Aditi, go.
When the glucose is too high.
Correct. So when you have high blood glucose, your body will produce more insulin because insulin helps you to metabolize sugar. It helps you to use or like promote using sugar. So when you need to reduce your blood sugar levels, insulin is produced. What's the quote unquote, what's the opposite of insulin?
Glycogen?
Close.
Glucagon.
Glucagon?
Glucagon, good. Glucagon,
Insulin.
which then promotes the formation of, oh no, sorry. Promotes the breakdown of, I didn't hear who said that, but how do we store extra glucose?
Fat.
Fat, but before that,
Glycogen.
somebody just said
Glycogen?
glycogen, right? Glycogen. Previous videos, right guys? Right. Glucose high, release insulin. Glucose low, release glucagon, right? Break down stored glycogen, release insulin to store excess glucose as glycogen. Bam. Okay. So insulin is a protein. It's a peptide-based hormone. Later on in the fall, when we do our options, if we go with option D, we will see sort of a bit more detail about protein-based hormones. But I mean, we know quite a bit about insulin already, right? We know that it's secreted by beta cells. How do you remember? Because there are alpha and beta cells in the pancreas, because insulin and beta is IB.
Ew.
IB. Glucagon is produced by the alpha cells because glucagon has an A in it. That's how you remember. That's all you need to know, okay? Know the basic function of insulin, which we have learned before, and we saw in another lesson as well. All right, let's keep going. What is this?
Immunoglobulins.
What does that mean? What is the word that we know?
Antibodies.
Yeah, and what is it for? Or what are they for?
For viruses.
And
To
what's the general term for bad stuff in our bloodstream?
pathogens.
Pathogens, good. So antibodies are also proteins. Actually, I'm going to send you, I'll try to send you a part of an article that I read today, which is quite interesting, that said that in Shanghai, they're treating some patients with plasma. It's like a plasma treatment. So people who have recovered already from the coronavirus, they're donating their blood when they leave.
Yeah, I've heard about that one.
Yeah. So because their bodies have produced the antibodies, the immunoglobulins, so we can use those to give to other people. In theory, it has a pretty high rate of success, so that other people can potentially use antibodies produced by a different person to fight their own infection.
How long does they reproduce?
Does what? What do you mean?
Right, how do the antibodies reproduce again?
So this, when we talk about vaccinate, like if you remember from last year when we discussed vaccination, so antibodies are made after the infection is detected, right? So you need your white blood cells, the lymphocytes to detect the pathogen first, then they start making lots of copies and releasing them and fighting the infection. It can take a long time though, because you're looking for a very specific lymphocyte, if you even have that lymphocyte that can produce the correct antibody. So there's not really a, I can't really say that there's a set time for that to happen.
So those patients that donate their blood will get infected again?
It's possible, but the second time you get infected, you will have what's called memory cells because you've already fought the illness one time. Then you will there will be more of those white blood cells ready to like jump to action. So anytime you get infected with an illness, it takes a long time the first time for your body to fight it. But then once it fights it, it creates like a backup, like lots and lots of backup cells for the next time. So if there's a second time, your body fights it off really quickly because there's already many, many lymphocytes ready to go.
Is this called passive immunity?
This is called, yes, your own response is passive. Yes. Yes.
I remember the virus.
We have a whole topic about this coming up someday.
I remember that antibodies doesn't work for virus.
They say again?
I mean, like I heard that antibodies doesn't work for virus.
Antibiotics don't work for viruses.
But not antibiotics.
Those are two different things. Because viruses are not living things, right?
Damn it. I ran out of food.
Okay, so yeah, just a little bit of detail about the antibody. I mean, everything we just said is basically what you need to know, right? They recognize specific antigens, which are proteins on the surfaces of cells, and then they help with our immune response. I'm going to keep going just in case it cuts us off at 40 minutes. I'm a little worried. Rhodopsin, this is one new one. Okay, rhodopsin is a protein, it's a pigment protein, kind of like if you imagine like chlorophyll is a pigment protein. This is a pigment protein in your eye, okay? And it helps with the absorption of light, all right?
Is it like cone and the cone and rods?
It's in the rod cells. So if you read here, it's in the rod cells of the retina. You don't need to know like the specific details of its mechanism, but just know that that's what it's there for, okay? It like, once it receives light, it changes shape and then changes to opsin. So it goes rhodopsin, changes to opsin, and then sends like a impulse through nerves to the brain, like basically saying that it's detecting light. Okay, collagen, I think we're familiar with. That's a really gross picture when you look at it like this. They are very long fibrous proteins, right? They're kind of like a rope. We see it in skin. We see it in our endothelial cells like blood vessels and ligaments and things like that. Also teeth, bones, okay? They're there because they can withstand a lot of kind of impact, all right? And I should look into this more, but I'm pretty sure that like if you go to the store, I mean, not now, but someday, you can see like a lot of like beauty products that say, oh, collagen, there's collagen in this. I'm not so sure that those are real. I don't think those work the same way that just forming your own collagen does. I'm skeptical, and I'm pretty sure I'm right, but it's like the new fad now.
Why would there be fat in beauty products?
No, fad, F-A-D.
Oh.
Okay, lastly, you've got spider silk. So this one is kept out because it's not produced inside of us, or it's not produced in the human body. But just some interesting points about spider silk. It has a lot of strength and tensile strength, and as well as flexibility. I mean, this is pretty cool. Stronger than steel, tougher than Kevlar. You can see the arrangement is different. So it's very like stacked on top of each other in the polypeptide chains. But some of it is disordered and can like come together when it gets pulled, which is why it has that sort of a lot of strength and ability to stretch and extend. But yeah. That's another example of a protein.
Wait, Mr. Nguyen, spider silk is different from spider web, right?
No, that is what it is.
Oh, but then how is it stronger than normally, you know, when you clean your house and the dust and then like you can just simply wipe out the spider web.
the idea is like if you were to
What?
build, like if you were to make something, like if you could
Oh,
explain.
okay.
like the similar properties, like if you make something out of the stuff that spider silk is made out of,
Okay.
but yeah, the intramolecular forces are
Oh,
stronger
okay.
than. All right.
Okay.
I hope this doesn't cut off in the next 10 seconds, guys. Let's pray that it
So
does.
if we take spider silk, a lot of spider silk, and make like a thick one, it's basically stronger than steel.
That's kind of the idea that they're getting at. Yeah.
So if we build a house with silk,
Yeah. Look at
Greenland would be
my
much easier.
silk house. Pure silk. Okay, so that's all for the examples, guys. All that's left is this. Okay, this is a very, very like up to the minute sort of research here. Is this idea of something called a proteome. We all know the genome, right? We have our genome, human genome, any organism's genome, like all of the DNA that makes up one, like what's in the nucleus of one cell. That's your genome. That's how what we can read, and then we can create proteins out of those instructions. But now this new concept is this idea of a proteome, meaning all of the proteins that any given organism that can make. If there's like this yellow box is yellow because this is the part you need to know. And the idea here is your genome is identical to every single human, with obviously a few little, like 99.99% similar between humans. But your proteome will be very much affected by the environment. So it's kind of more like the phenotype, right? Based on what your experiences are, what happens in your life, your proteome could be different to another person, right? Like at different times in your life, different stresses, different whatever experiences you go through, your body will adjust the proteins that it builds. If you take a simple example of, let's say, like skin pigmentation, right? Like if you happen to move to, or let's say you move to like a very, very sunny region of the world, like your body will probably eventually, if you stay there for long enough, eventually start to produce a greater amount of like darker skin pigment to help you deal with the fact that you're always in the sun, right? Whereas if you had a twin, an identical twin who's genetically identical to you, and they stayed in a region that didn't have that much sun, their skin wouldn't change the same way. But you guys have identical genomes, but your proteome adjusted and diverged based on your environmental factors. So this is like a cool area that people are studying now, is to see like how can we look at a proteome and see what benefits can we get from it. How can we maybe treat people differently based on the proteins they produced, in addition to just looking at their genetic makeup.
Oh.
It's pretty interesting area. I'm just gonna.
Podium can like forever like vary by time, like for each individual due to environment change.
Yeah, exactly. So like the best way to think of it is like identical twins, right? So identical twins have the exact same genome, but if you put them in two different situations, like their proteome will end up adjusting to what happens to them in their life. So like different genes may get turned on and off depending on what happens to you throughout your lifetime. So like genome might be absolutely identical, but proteome could be something that changes a little bit over time. Later we'll.
Is proteome genome?
Well, proteome is based on genome, right? Because to make a protein, you need the instructions from the DNA. So the genome is your DNA, right? Whereas the proteome is the proteins you build from that DNA.
Okay. So proteome is a product from.
Correct. Yeah. So essentially, this is really just the central dogma, right? DNA to RNA to proteins. But what they're saying is if everyone starts with the exact same DNA, it doesn't mean that we end up with the exact same proteins. We can all make the same proteins, but we won't necessarily make the same proteins because things happen that require like different production at different times and different amounts and so on.
Okay.
Yeah.
Ah.
Okay. We'll finish off here, guys. Oh, yeah.
So fast?
That's so fast.
Yeah, this is a quick one. No, sorry, there's a little bit more. I want, if I'm gonna see if this works, try to take a video of it if you can do it. This will come up again when we do enzymes next lesson tomorrow a little bit, is this idea of denaturing. We all know this term, denaturing of protein, right? And what are the two things that we studied in IG that can denature protein?
And
temperature?
Temperature and no.
Yes, temperature is one.
And pH level.
pH level, right? pH level. Okay, so the same thing, right? Here you have two boxes. Heat can cause denaturation. Extremes of pH can cause denaturation. What we want to be familiar with is that usually denaturation is permanent, meaning, have you ever tried to unfry an egg? Right? Once you fry the egg, it's fried. You can't unfry it and make it.
But which one tastes the hen or the egg?
Right? Like you can't like cook an egg and be like, oh man, I wish I hadn't cooked that egg. Oh, I'll just uncook it and put it in the fridge. No. So most of the time, denaturation is permanent. Okay, that's it. Once it's denatured, it's over. Because the shape of the protein has changed too much. They've broken too many bonds that cannot be reformed. You've kind of like impacted the fragility of this molecule. And of course, like enzymes is the key one that we'll look at later. Okay, so because of these bonds and the interaction, whoops, I forgot I was drawing. Because these interactions are quite weak, okay, they can be messed up by extreme heat. They can be messed up by the pH being either too acidic or too basic. Why? Because you're shoving in a whole bunch of like positive charges or negative charges, depending on what you're talking about. And they're going to make new bonds and just mess up. Like we have that nice blob shape, whatever it needs to be, but then suddenly it starts to fall apart and now it doesn't do what it was meant to do initially. So one thing you could try is, and I don't know how well this will work, I'm gonna skip. This is just telling you that there are proteins that exist in very extreme conditions, like the near like hydrothermic vents, which are extremely high temperature, but they're still living things down there because species adapt. So they have proteins that are able to withstand high temperatures, higher temperatures than what we would be able to withstand. So if you're feeling ambitious and you have an egg to waste, you could try this experiment at home, where you try to find, and you need a thermometer, which maybe you have, or you can borrow your security guard's thermometer, is to try and like basically take the white of the egg only, not the yolk, and to see like at what temperature does it start to denature. So you'll know because the color will start to flip to a solid white. In a perfect world, we would do this a whole bunch of times and consolidate all the data and find the temperature stability. At what point is the albumin or the white of the egg stable, and at what point does it denature? And that is the end of that topic.
I don't have eggs.
I'm gonna shut this. Hold on a second.
Don't waste your eggs. Come on. There's like no eggs in Shanghai. And it's so hard to get eggs here.
I still those times.
What? Really?
It's
Yeah,
so
like
easy to get
my
eggs.
mom has to go on Hema and in the morning, 6 a.m. to click egg and buy it. Otherwise, it's gonna be sold out all the time because
But like if you only go on Hema,
Okay, wait.
go on other apps.
Yeah, she does go on other apps, but like that's where we go to and stuff.
Okay. Before we get on the egg thing, does anyone have any questions, either from this lesson or anything we've done up until this point, that you'd like to ask while we're all here?
I think that means a no.
Sweet.
His kitchen looks like a science lab.
It kind of looks like our science lab. It has the same color.
Okay, so we're
You
just
live
gonna,
at SSIS. All right, I'm gonna stop this recording so that it's finished.